Please, yes. Are you there? So, good afternoon once again. Just like I told you that this is another opportunity for us as bioinformatician to look at the general workflow if we want to work on bioinformatics as our um, project. Now, let me try as much as possible to introduce to you what we want to do for the next few days. I told you that it's going to be in the next 5 to 7 days that we are going to do this. So we want to look at the general workflow when it comes to the issue of what do we mean by workflow? That is from the first step to another step to another step to another step. Don't forget I said it's going to be from wet lab to dry lab. So what do we mean by wet lab? Anybody with wet lab? What do we mean if we say wet lab? Okay, any idea? Wet lab. What is wet lab? Huh? What is wet lab? Talk. You said lab work. That is the one that is going to involve you culturing, isolation, and identification. So we want to start away from when you are collecting your sample. When do you want to collect your sample? We want to start from there. That is from wet to the dry. So the first step that I want you to know, I want to typically give you an example of somebody who is working on TB, drug-resistant TB, who is trying to start from collection of sample to the last one and is going to design vaccine using bioinformatics. So the first step is going to be number one, what we call pre-sampling. Pre-sampling. Pre-sampling exercise, that is the one that is going to include that person must first of all identify who are the stakeholders, who are the major stakeholders in his research, major stakeholders in his research. So, and who are those ones that we call stakeholders? That is, those that are going to be too relevant for him. You want to collect sample, you want to collect blood sample or sputum sample. Don't forget that because the person is working on TB, is going to be interested in which type of sample. Is it blood, saliva, urine, or what? So because Mycobacterium tuberculosis is an airborne disease, it is an airborne disease, the person will want to focus on sputum because sputum has to come directly from the lungs. So you will talk about sputum. So you have to identify those that are stakeholders. Some of these stakeholders include the nurses. They include the governments, hospitals, private hospitals. the management of each hospitals, the community. The community head, there are times that part of your pre-sampling will be that you have to engage them one-on-one, tell them what you want to do, how is it going to be too helpful for them. So that's what we call pre-sampling. The next one has to do with advocacy stroke consultation. Advocacy means that you are trying to convince those stakeholders. You want to convince them, you want to educate them on what you want to do. You want to educate them. So that is what we call advocacy stroke what? Consultation. Then another thing, apart from, don't forget that identification has to do with in those communities you are interested in. Consultation has to do with those you are also interested in dealing with outside. Then another consultation is that you have to also consult with your supervisor. You consult with your supervisor approve the topic for you, you carry him along, you tell him what you want to do, the objective of your work, what you are about, why are you so interested in that work. It might not really be in Mycobacterium tuberculosis, it can be another. It can be maybe you want to work on cholera, you want to work on typhoid. So all these are part of engagement with your supervisor. Then under pre-sampling, the third aspect has to do with preparation of research instruments. Preparation of research instruments. For example, if you're going to make use of consent form, because you are going to collect human sample, you need consent form, you need their consent. You will produce something that, you produce some document that is going to educate them on what to do, on what to expect, where you are going to spell out the real advantages of your work, and you also tell them the risks as well. So it's part of it's what we call the consent form. So, and if you want to talk about consent form, that should be a place, because before you recruit the person, you cannot just go to the community and just tell them verbally that I want to collect their sample, I want to know. There should be a place whereby they will consent, they will agree that they want to be part of that your study. So show recruitment form, you will recruit them. So where they indicate consent, where they indicate that they are interested in it, that is a recruitment form. Okay, then you need to also get, because you are working with human sample, you have to get what is called the ethical approval. The ethical approval, we have a committee that is actually set aside for that's going to review all your research documents and will be able to tell you whether you can proceed or you may not proceed. That is that. Then you recruit some workers, field workers, people that will assist you, because you are not a trained medical professionals. You need, or medical personnel, you need somebody that will tell you that, okay, they are going to help you and assist you in that. Okay. Another thing, apart from that, will now be that will be the end of your pre-sampling. That will be the end of your pre-sampling. Don't forget, the pre-samplings are made up of how many sub items? Five. The first one is what?
Identification of stakeholders.
Identification of stakeholders. Two.
Advocacy of concentration.
Okay. Three.
Preparation of research instruments.
Four.
Ethical background.
Okay. Five.
Recruitment of workers.
After you must have done that, you now proceed to the second aspect, which is going to be collection of samples and transportation of that sample to your laboratory. you collect. How will you collect from their community? Maybe if you are recruiting them from hospitals, how you will collect their sample is very, very important. The instruments you are going to use in collection, as well as how you are going to transport them to the laboratory. Transport them to the laboratory, okay, is very, very important. The method of collection determines what is going to be your final results. So that is a collection and transportation of samples. The third one, don't forget that I told you that we are using a case study of somebody who is working on what? More, that is what? Drug resistant, or what you can call Mycobacterium tuberculosis. The person,
The video is blocking.
the person's how the person is going to be, you have to manage it that way. We are going to let me see if I can adjust it from here. You said it's blocking what? Okay, let's see. Okay, I think I've adjusted it now. Okay, now check if it has been adjusted. Check now. I think it's right. It must have been adjusted. Has it been adjusted? Refresh before we move on. Has it? If it has been adjusted, let me know. Refresh. How about it now?
Yes, it has been adjusted.
Okay, so it has been adjusted. Okay, now as I was saying, the fifth, I was using an example of somebody who is working on Mycobacterium tuberculosis. Not even somebody, that is my own work. That is my own work. So I'm just making use of those. So after collection and transportation of that sample to the laboratory, the next one is to start processing of that sample. So processing of that sample will now determine what you are working with. Don't forget, those that are working, the lab workers, they will follow the same procedure. That's when you see them preparation of media. Apart from preparation, they inoculate them and so on. But in case of Mycobacterium tuberculosis, I think 4 important processes are highlighted here. The first one is the contamination of that sample. The contamination of the sample, then you run through by the use of GeneXpert machine. So you do what is called GeneXpert. So GeneXpert also work with is part of the first step in DNA analysis. It's a DNA analytic machine. So at that point, under GeneXpert, it is going to, for somebody that is working on Mycobacterium tuberculosis, is going to tell you whether the person has sensitive TB or whether the person has resistant TB. It is at that level of GeneXpert that you will know. So it's going to classify it into S, which is sensitive, and R, which is drug resistant, DR. So after that, you will now do what is called smear microscopy. Smear microscopy, that is what we call acid fast staining techniques. The use of xenazine stain. Okay, so you use acid fast. Don't forget that Mycobacterium tuberculosis is an acid-fast bacteria, bacilli. That's why they call it AFB. It is different from, it is not gram-negative or gram-positive. Don't forget, we are going to, if I do not give you all this background, you might not actually know where you are going to. So even if you are going to do dry lab work, if you do not have, that's why I said if you want to use bioinformatics, you also need to know about the biology of the organism you are dealing with. If you don't know the biology, if you don't know the wet laboratory analysis, you might not be able to actually understand it. And don't forget that this bioinformatics group, I have divided it into three. The first one is an antimicrobial resistance group, AMR group. The second one is vaccine designer group, that is in silico, those that will use vaccine to be able to design, that will use computer to be able to use it for what? Okay, so the third one has to do with drug design group, that is those that will design drugs that can be used to control the. So even if you are retrieving sequences, if you just say you want to only do dry lab and you want to start with retrieving your sequences, many of you, from my interaction with you so far, you don't know why. You don't know why you are retrieving. You are just telling me you just go to NCBI, you just know. If you are going to NCBI or you are going to any database, what are you going to retrieve there? Why are you retrieving it? If you don't know what you are, the sequences you are to retrieve, because in that NCBI, you have protein, you have gene, you have genome, you have nucleotide, you have a lot of them. So if you want to retrieve, which one are you going to retrieve? So that's the reason why you need to also have the wet lab knowledge of all this. So immediately after the smear microscopy, you now talk of isolation and culture. Don't forget the essence of isolation. The sample, the organism you are looking for, they exist in community. They exist as with interrelationship with others. It is not only them. So you now want to isolate them. It's just like, for example, now you are looking for a particular, okay, let's say you are looking for a particular girl who is Rachel by name. If she is Rachel by name, among all the people in your class, there must be a reason why you will get Rachel on her own without dealing with others. That means you need to subject Rachel, identification of Rachel, into different. Okay, is she dark in complexion, tall, male or female, and so on, like that. That is part of isolation. So the wet lab scientists, what they have been trying to do is to isolate that microorganism from the sample, and that's why this one takes a lot of energy. And if you do not use some specific method, if you are not so passionate, you might not be able to isolate Rachel. So you just isolate Rachel with some other people, whereas you are not interested in other people, you are interested in Rachel. So if you want to get Rachel, you must produce something that will only single out Rachel from among all other. That is what is called isolation. So isolation, you can use culture media, you can use biochemical tests, you can use a lot and a lot. Now, the end, that is now the isolation and culture now spell the end of wet lab. At least at this level. So the first one, at this level, we spell out the end. The other part of it, of wet lab, is going to be us. After you must have isolated it, you are going to keep your sample in a bottle, and it is going to be transported to where they will continue with molecular analysis. Don't forget that you, as a bioinformatician, what you are interested in is to get your sample in form of alphabets, and the alphabets are going to be in form of a nucleic acid, which can be in form of a DNA or RNA. It can be in form of DNA or RNA. Now, that will now spell the under isolation. I've mentioned biochemical tests, a lot of them. So it's still under wet lab. So number 4 now is molecular analysis. After pre-sampling, followed by collection and transportation of samples, followed by sample processing, the next one, the next stage is going to be molecular analysis. And molecular analysis, we always start with DNA extraction. You, out of the whole organism, what you are interested in, because don't forget that nucleic acid, which is in form of DNA and RNA, we call them informational molecules. These are the ones that can give you every information about your microorganism, because it is this DNA that determines their gene, that determines their behavior. If they are virulent, for example, if COVID-19 now, coronavirus, they are virulent. If you want to know why they are virulent, they have molecular basis. If it doesn't have molecular basis, we might not in any way actually understand them. So, and under normal circumstance, if you do not understand the molecular basis of any microorganism or any organism at all, it might be too difficult for you to even control them. It might be difficult for you to control them. That's why, because you want to know, because you have information about about them at molecular level, that's where you can now come and say, me, I am interested in designing vaccine for them. Me, I am interested in designing drug for them. Me, I am interested in knowing the drug resistance, the antibiotic resistance with them. So all behaviors has molecular basis, and that's the reason why we continue that way. So the first step is going to be DNA extraction. After that DNA extraction, the next one, which I did not put, is what is called polymerase chain reaction, PCR technique. What does polymerase chain reaction does? Number one, what PCR does is to amplify. PCR, you just have very small segment of that DNA. But by the time you now have that small segment, how can you magnify it? How can you amplify it so that you can have it in multiple copies? That is what is called the polymerase chain reaction. Polymerase is an enzyme that is also found in bacteria, but not ordinary bacteria, bacteria that live in extreme high temperature. So polymerase chain reaction, you now use that enzyme to be able to amplify, to get multiple copies, to get millions of copies of the organism you are interested in, so that by the time you now want to do whatever you want to do, you will now do it. So after the DNA extraction and PCR, the next thing is library preparation. Library preparation. You know what they do in the library? In the library, you have a lot of books. That is where um, um, information is being kept. So from that library, so what you will now do is that I want you to look at me. Let's say this is the microorganism which you have isolated. In this microorganism, you have now gotten the DNA. What you are interested in is the DNA. By the time you get the DNA now, which is the informational, which contain all the information about that organism. And in some of my teachings before, I have told you that once you have an access to the DNA of any organism, you have gotten access to everything about that organism, because it is going to give you a lot of information about that organism. Ability to resist different antibiotics is in the DNA. Ability to affect or cause death, it is in that DNA. Ability to trigger an immune response, it is in that DNA. So everything so far depends on that DNA. So immediately after that DNA, don't forget that the DNA will be in a very minute quantity that you might not even know. By the time we put out, you see, what we call the DNA, you cannot see it with your naked eye. Microorganism, you can't see it with your naked eye. Even when you have extracted it, you can still not see it with your naked eye. What we are saying is that if it is going to take, if a certain microorganism is on 1,000, that is, you have to look at under the microscope, you now is going to take you 1,000 until you multiply or until you magnify that microorganism in 1,000 before you be able to see it, if that is microorganism. Now, to be able to see the DNA, is it going to be of higher magnification or is going to be of lower magnification? For the DNA, where do you find the DNA? It's found inside the microorganism. The organism is visible by 1,000 times. So if you want to get the DNA, that means it's still going to be more than 1,000. It could be maybe after 10,000, after 100,000, before you'll be able to pick the DNA. Now, that DNA that is now very, 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 very small, minute, you now want to amplify it. Let's say you have let's say you have this is the DNA. Please look up. Let's say this is the DNA. This part of what you're supposed to learn in microbial genetics, where we call it gene regulation. Gene regulation or gene expression. How are genes expressed? So on this straight line of the DNA, it could be just one portion. It's not everything. For example, it's going to be from here to here. Could just be what is responsible for the virulence. It can just be out of 1 million or 5 million nucleotide bases, it can only be maybe 5 only nucleotide that is responsible for drug resistance. So that means you need to amplify it, you need to magnify it in so many times. And one thing you should know is that any gene that is being expressed by microorganism is an essential gene. It's very, very essential. What you normally produce is what you needed most to be able to survive. So when you now amplify it with a PCR, so the next one is library preparation. Library preparation is that you have gotten your DNA now amplified. You are now looking for it inside the library. It's like when you have a magnet and you are looking for a needle in the sand. Anytime you scan through the sand with your magnet, if the needle is there, it's going to bind to the what? To the needle. And you'll be able to now take the needle out. That is what is called library preparation. It is like a magnet. You are looking for a magnet. You are looking for a needle in sand. So you need a magnet. So when you must have now gotten your what? Your needle now, the next thing is now going to be two things: sequencing. Sequencing means you now break the DNA, you now break it down into alphabets. If you are talking of DNA, that is what is called the nucleotide. If you are talking of DNA, there are 4 different types of alphabets in DNA. A will bond with what? T. G will bond with C. So, and that's the reason why when you have a lot of sequences, you'll be able to differentiate any one. If it is nucleotide, if it is DNA, you should know. And if it is RNA too, you will know. So that is that. Then after that, apart from that, there are now 2 types of sequencing. Sequencing, number 1 is called 16S ribosomal RNA. That is what is called partial sequencing. Partial sequencing. Are you getting me?
Yes.
Partial sequencing. So partial sequencing, I cited an example of looking for Rachel for you. So if you want to do partial sequencing of Rachel, that means we just want to identify Rachel with just one finger. And don't forget that the finger of Rachel will be too different from every other human being. So that is partial. We are only looking for 16S ribosomal RNA. That is partial. So, and you will not get as much information about Rachel when you are using the finger alone. So that is what is now going to be the next one, which is whole genome sequencing. Whole genome sequencing means that everything about Rachel now, we are no longer interested in the hand, but we are interested in everything complete. So we will be able to know that, this organism has these types of gene. The drug resistance gene, there are 5,200. The whole alphabet that is contained in that organism is 5.5 million. That is what is called whole genome sequencing. Now, for some of you that have started your work and you have started retrieving your sequences, which sequences are you retrieving? Are you retrieving sequences from ribosomal RNA, which is from partial sequencing, or you are getting the sequences of the whole organism? For example, now, if you want to see, look at the antimicrobial resistant gene in a certain microorganism, it is good for you that you get the whole genome sequencing, the totality of that. So, but some of you, what you have done so far is just that you just go and retrieve the sequences in pieces. For example, now, look at what you have been doing. You want to develop you want to develop a vaccine. What is your name?
Like,
Olaitan. You want to develop, you want to know about Olaitan. You want to know about Olaitan, you want to learn more about her. So you now get two information about Olaitan. You get 3 from another person. You get 4 from another person. Forgetting that A, B, C, D, they are different, although they are human beings, but they are different. So what we are saying in essence is, when they say Mycobacterium tuberculosis, there are different species. You now go and get, for you to be able to prepare your own protein, you are now getting, you get 5 amino acid from strain Abeokuta. You get 15 amino acid from strain Kenya. You get 5 from, so it's quite different, although you are working with that strain, but you are not designing vaccine against one person. Are you getting me? You are not designing vaccine against one person. So if you want to design a vaccine, it is better you use whole genome sequencing. And if you are using whole genome sequencing, that means you will know that the strain you are producing vaccine for, or you want to see antimicrobial resistance, it is only one strain. And you can now from there generalize it. Instead of you to pick from this, that one, and that one. And at the end of the day, you are not going to get a specific. And don't forget where we are going to. Don't forget one of the reasons why bioinformatics is the bride of the day, and is the bride of some years to come, is that we are working towards personalized medicine, personalized vaccine that will no longer be useful generally, vaccine that is going to be drug that is only going to be meant for you and no other person. If you have any side effects from your drugs, it is going to be that side effect for you only, not for another person. Pharmaceutical companies, they are already changing their narrative. They are already changing their method of production. They are no longer producing drug for general population, because they have discovered that most of the drugs they produce now, and microorganisms are now developing resistance towards it. And one of the reasons why they are developing that resistance is as a result of misuse and abuse, non-prescription. They will just take it anyhow. And that is why the microorganism, the antibiotics that were very useful to cure your ailments, to control your diseases in the last 3-5 years, they are no longer effective now. And that is why WHO now declare that, for example, now there are some bacteria, there are some organisms that they have developed total resistance to all drugs. In fact, I'm very familiar with that of a Mycobacterium tuberculosis. Mycobacterium tuberculosis, they have 3 or 4 levels of resistance. The first one is monoresistance. That is when they develop resistance to any one of all the antibiotics. That is mono, but it has to be single. We also have some that are called drug resistance. That is when they develop more than only 2 of the antibiotics that they use in controlling them, they are no longer useful. They are no longer effective. That is drug resistance. But we now have what is also called HDR. That is extensive. Let's say there are 10 antibiotics that they normally use to control them. If 8, they are now resistant to 8, what is the fate of the 2? So that means they are HDR. And nowadays, we have, since 2012, they have also reported what is called TDR, total drug resistant, that no amount of antibiotics that you use in controlling them. We now have some strains of Mycobacterium tuberculosis that they cannot develop sensitive. They cannot be sensitive to any antibiotics any longer. So that is what you are looking for. That, okay, it is better we use computational analysis. It is better we use our computer to be able to, even before they will produce, even before we will now tell the pharmaceutical companies that they should go and produce antibiotics, can we predict? So that is AMR, that is drug resistance. And the drug as well, we will also see this. Now, let's now go to the dry lab, so to say. The dry lab, so to say, and which we are going to start now. Please, I don't know if you have any question. If you have any question from step 1 to 4. Step 1 to 4. Now, one thing I want you to know very, very well, which I have been telling you, is that ChatGPT should be your closest companion. I've been saying it on a lighter mood. I've been saying it. Your supervisor does not have all the time, and sometimes even the knowledge of your supervisor can also be what? Limited. If your supervisor has not actually worked in the organism you are working in, the supervisor can only guide you. So, but ChatGPT is going to be the one that will assist you a lot. And that's the reason why I said, let it be your very closest companion. When others are not there, let it be only you and ChatGPT. During the dry and during the wet. If it is convenient and when it is not convenient, let it be your closest companion. If you don't know how to use it, please go and get YouTube, watch YouTube on how can you make use of ChatGPT for your research. Gone were those days that you crack your brain to be able to get a lot of information, for you to be able to design topics, for you to be able to design what are the trending topics in your field. In bioinformatics, what are the trending topics in vaccine design? ChatGPT, if you ask ChatGPT to give you 50 or even more topics, it's going to tell you. And you start picking it one after the other. That's me. I don't know anything, ChatGPT. I want you to assume that you don't know anything. You are a student that doesn't know anything. So, and you want to work on this and this and this. Can you please guide me? Give me the steps. Give me everything. What have I done right? In fact, I was even telling those in the lab workers, gone were those days that you will strain your eye, you do a lot. When you have done your isolation and you want to read your plate, nowadays, just snap it. Snap the plate, attach it on ChatGPT, and tell ChatGPT to identify it for you. It's going to do it accurately for you. So if you don't know anything at all, what you should allow to be your closest companion should be what? ChatGPT. And which is very, very important for you. It is very, very important for you. So let us start with, don't forget that I said that after this training, you are the one. I'm going to demonstrate each of these tools. There are some languages that you should get used to them. if you are going to work on what we call bioinformatics. It's also called computational biology. It's also called computational biology. And we are going to make use of, I told you that most of what we are going to be doing, we are going to be making use of ChatGPT from time to time. So when we get to any point and we seem to have lost the road, we have left the road, we don't know what next to do, please and please, ChatGPT is your closest companion. Ọ̀rẹ́ nígbà òtítọ́, when others have forsaken you, when you seem not to have anybody that can come to your rescue, ChatGPT is always there, and it is going to really help you to be able to do a lot. You are going to get a lot. So now let us now imagine that after the first processes, you now have sequences now. After the molecular analysis, DNA extraction, PCR, library preparation, and you now have whole genome sequencing. Okay? Yeah, please open your laptop now. Open your system and go to, I gave you I think I sent a link. I sent you a link of Galaxy. Abi? Okay. Galaxy. So, have you registered on Galaxy? Some of you that day, some of you said you have registered. Though. Yes. So just go to your Are you there, Galaxy? So we want to start. Go to Galaxy. Open your Galaxy. Check Galaxy now. Can you see it? It's going to be displayed on your system. Where is the one you registered? Say you should register. Go to that link again. Go to the group chat platform. Go and see when I said you should register. You see, some of these things, even if you don't know, the site, oh yeah, go to ChatGPT, open ChatGPT, tell ChatGPT to give you the link. You want to analyze, you want to use whole genome sequencing, so go to ChatGPT, open it. This your system that is taking 2 years to 19. What's the model of your system? 19. Okay, I think it's Galaxy. Look at, you've not registered. It's not going to take 24 hours. So it's better you register now so that you just watch this one by this time tomorrow. Go to the ChatGPT. So if you don't know anything, go to ChatGPT. Open it. Let's see. That this is Google. So this is taking 2 years to load. ChatGPT, okay, link for genome, okay, all for GS. Let's see. You can see the fourth one. So you can see, even if when I said even if your supervisor forsake you, yes, sir, ChatGPT cannot forsake you. Okay, link for genome, okay, original GS analysis. So you have to register first. One other thing is you have to be used to your this thing. Where is the link? Regenerate it. Link for whole genome analysis. I told you to have signs. Please not to. Okay, please go back to Bio-Nexus. Go back to Bio-Nexus and watch me on Bio-Nexus. Go to Bio-Nexus. Are you there? Are you there? Please watch me on Bio-Nexus. If you are there, raise up your hand. Okay. Only few people are there. Put your system on Bio-Nexus. If you are there, raise up your hand. Put your system on Bio-Nexus. Only few people are there. Put your system on Bio-Nexus. If you are there, raise up your hand. Put your system on Bio-Nexus. So, watch me. Watch me. So now, I want you to know. Don't forget, where did the molecular analysis, where did it terminate? Where did it end? Molecular analysis. Yes, give me the techniques under molecular analysis. Can you give me those techniques? Put your system on Bio-Nexus. Library preparation. There's one you are missing before library preparation. Watch me. Now, whole genome sequencing. Please listen. Whole genome sequencing. Watch me. When the sequencing machine. Now, I want you to know. When the sequencing machine. Don't forget. There are two. Where did the molecular analysis? Where did it terminate? Where did it end? There are two very much important yes, give me the techniques under molecular analysis. Can you give me the first one? You can use what is called Illumina sequencing library preparation and nanopore. You are missing before library preparation. It used to be Sanger sequencing. Now, whole genome sequencing. Sanger sequencing. Listen. We are going to learn this in molecular genetics. the
Sanger
sequencing
sequencing.
machine.
I want you to know.
We have
When the sequence,
I want you to know.
sequencing machine.
And we also
Don't
have,
forget,
there are two,
where did the molecular
there are
analysis,
two
where did it
important,
terminate? Where did it end?
There are
Molecular
two
analysis.
very much important
Yes, give me the
techniques.
sequencing.
Hello? Are you getting me? I said there are Sanger sequencing techniques, Illumina sequencing, Illumina sequencing, and we also have what is called nanopore. Nanopore. So I want you to give me, I'm going to give you this as an assignment tomorrow. Okay? I want you to differentiate. You have to enumerate, enumerate and differentiate between all sequencing techniques. All sequencing techniques. All sequencing techniques. Describe in details. Describe in details the mechanism by which they operate. Describe or discuss the mechanism by which they operate. Then the rationale. What are the rationale behind what are the rationale behind what are the rationale what are the rationale behind the choice of each, the rationale behind the choice of each. That is, why do you choose anyone that you want to use? So let's go to watch me as I proceed. So if the way it is going to come out from the sequencing machine, your data, your DNA whole genome sequencing is going to come out in form of FastQ, F capital letter FASTA Q, FastQ. Okay. Now watch me now. What I want to do, okay? Let me just first of all highlight what we want to do. The step that I want to do. Number one, watch it on YouTube. Number one, the project setup. So I told you that the whole genome sequencing is going to come out in form of FastQ. Okay. So any data at all, the first thing you need to do is to clean and organize your data. Clean, because the sequencer, that is the sequencing machine, it is not 100% efficient. There could be contamination, there could be a kind of a mismatch, error, different types of error. but the error is going to be very, very minimal. So the first thing you need to do is to subject it to what? Cleaning. You have to clean your data first of all. You clean your data. So, and if you want to clean your data, now one of the reasons why I want you to master the use of a Galaxy, there are different types of tools on Galaxy that you have many just in one website. You have a lot to do. You can clean your data, you can annotate your data, you can assemble your data, you can join your data, you can convert your data from one form to another. There are lots. So when you look at, I don't know if you have seen this. Let's go to Galaxy. You will see it there. If see the Galaxy. If you are there, just raise up your hand. If you are, if your YouTube has opened Galaxy for you, has it opened it for you? Galaxy. Has it opened it for you? Hmm? Can you see this? Can you? Are you there? What happened?
Yes.
Are you there?
Yes, sir.
Okay. Now, if you look at the left hand side, look at the left hand side. Please look for my cursor. You will see upload. You see tools. Can you see it? Left hand side on Galaxy. Galaxy. You see upload. You see tools. You see workflow. You will see workflow invocations, interactive tools, visualization, histories, history multi-views, and you see notification and more. Can you see it?
No.
It's still there. Check. It should still be there. Are you on YouTube? But why? Why is it? Are you there?
Yes, sir.
Are you sure? Also, can you see all those things I said? Can you see them? Galaxy, upload. So from there, click on upload. Click on upload. I hope you are documenting it. The first one, I told you that after whole genome sequencing, that your whole genome sequencing, sequences, you will have it in a flash drive. You have it in form of flash drive because they must have converted it for you into what? Into data, into alphabets, into sequences. And I told you that it's going to be available in faster formats. So the first thing you will do is to clean that data, to remove. Why do you need to clean? So that you'll be able to remove contaminants, you'll be able to remove mismatch, error of mismatch, and other errors. So what you will do is you will now click on upload. Click on upload. Click on that upload. Have you clicked on it? Okay, have you seen it? Click on upload. Let me see. Okay, it's not yours. What's wrong with this? No light. You are not there now. Why? Go to YouTube. From the YouTube, that's where you'll be watching it. And then you just the steps are going to be taken. You are the one that will now be writing those steps down. It is when you get old, you now follow that step to do whatever I ask you to do. You are still on the YouTube. It is the YouTube I'm asking you. Yeah. And this is where we are now. Because you have clicked on upload. Upload. You have your file in form of FastQ. In form of FastQ. Your file is coming in form of what? FastQ. After that FastQ now, you just say this for you have to clean your data and to remove error of mismatch and so on. So you now upload. Click on upload. So you follow it. So when you click on the upload, you will see where we have choose local files, choose from repository, paste/fetch data. You will see start. You will see pause, you will see reset, and you see close. So where you will click, just click on choose local file. Choose local file. Choose local file. Already, I have my own sequence data. Whole genome sequence. I have my own sequence data. I already have it on the system, on this system. So, and it is on my desktop. It is on my desktop. It is on my what? Desktop. So, my own, don't forget that I'm working on TB. So, I click on TB sequences. I click on TB sequences and I want to upload. Now, I want you to take note of something. Take note of something. When you are getting your FastQ is coming in form of reverse and forward. Don't forget the DNA is two-stranded. It's a two-stranded molecule. So it's going to be in form of reverse and form of forward. So you will now upload both reverse and forward. So I want you to let me pause a bit. I want you to show me. Just still be on Bio-Nexus. You see Bio-Nexus. You are not following. I said you should not leave Bio-Nexus. Watch it. You can see this is 1, 2, 1, 2. So I click on. Yeah. These are my 2 sequences. 1, 2. So you can see. You are not getting what? You are not following. It's not like that. You are not following. It is when you get home. What I've taught you today, what I'm teaching you today, when you get home, you are the one that will now do the point at which we are going to stop. Huh? But if you are not getting it, if you cannot get it, you won't be able to do it on your own. Okay? Your own sequence. Don't do your own sequence. It's when you get home. Just watch me. Watch that. So when you get home, where you are going to get it. Now, I need to know. They are not holding on now. Complete genome. What is their? Follow what I ask. Okay. Once you are there. So watch me now. Watch me next. I'm going to click. You can see that it has brought the 2 sequences there. The first one, you will see that the first one is forward. The second one, the first one which is forward is 1. Can you see it? KWA_1, that is forward. When you look at the second one, 2, that is reverse. 1 is 182.5 MB. The second one is 188.3 MB. Now, after that, just go and click on Start. Start. So you will see it. Just note it. Kò ní click. Just watch it. You will see it. It's already rolling. It's moving. Oh. Yeah. When you now become masters now, you now be. Don't forget you are the one that will teach the HND one when they want to do their bioinformatics. So, so it's better you. So I want you to see the progress. Can you see the progress? The progress. You click for where? You start. You can't do it now. You see me, and you see I. It's not you that was. Now, don't forget you are the one that will teach agent one when they Okay, let's see. Can you see it progressing? So I would just want to say, like, the next 10 minutes we stop for today. So if we are stopping for today, what you will do is that you will go to NCBI, go and get, or you um go to ChatGPT. Tell ChatGPT to give you FastQ formats of any organism you want to work on, just for practice. It might not be the one you are going to make use of your projects. So ask it to you retrieve that sequence there, whole genome sequence, and I want you to upload it on Galaxy the same way I've uploaded it. And let us see how it is going to get uploaded on your that. Now, another thing I want to tell you is that if you look at what I've just displayed here, you will see the tools raw read QC. So we want to check the quality control. We want to clean it. We want to check how clean your data is. So that's why it is like that. So go check sequencing quality, contamination, and adapter contents. And the tools you will make use of is FastQC. This FastQC is used per sample. So it's going to clean it one by one. If you are using FastQC, that one is going to clean it one by one. If you use multi-QC, that means it's going to aggregate. That is, if you have more than one sample, let's say you have 10 samples now, we are going to make use of multi-QC. So that one is going to aggregate all the FastQC reports for you. And the procedure is as we do it and as you have seen it over there. Now, before we actually close for the day, I want you to know one thing. So I want you to open a file. Go and get a file, sequences from NCBI or from wherever. Get FastQ files of 5 different organisms, 5 bacteria, not 5 bacteria. You can see I've already given you like 3 or 4 assignments. The first one is different types of what? Different types of differentiate. The second one,
No, the second one is go to MBC.
and the second one is you have to demonstrate. That is, you have to upload what I have done for you today. You have to do it. Then the third one is that you have to retrieve FastQ format for 5 different bacteria. 5 different bacteria. So it is on that note, you can see the way my uploading is progressing. You can see the way it is progressing. Now it is around 44%. It's around 44%. It's going to do it one after the other. After it must have loaded up to 100. So the next one is going to take the stage, followed by the other one. So I really want to thank you for your attention today. Tomorrow we are going to stay longer than this, so it's because you do not